Hi everyone and welcome to the internet. So what we're going to be looking at in our lesson today are things called stem cells. So hopefully by the end of this lesson you're going to be able to describe what a stem cell is and explain their functions. To start us off though, we've got 3 questions based around our previous lesson, so have a go at answering those in your book. So I'm just going to give you a few minutes to have a go at that. So let's check those answers then. For question 1, I've given you the ones we looked at in our previous lesson. There's actually way more than that in terms of cells in our body that are specialised. So up to 3, give yourself a mark for each one you've got: palisade cell, sperm cell, red blood cell, fat cell, ciliated cell. You could have gone down the route of saying things like nerve cells, white blood cells, the list goes on. Number 2, we had to explain 2 of these features. Now, where it's an explain question, that means we need to say what the feature is and how it's actually helping the red blood cell. So to get the mark, you've not only got to say what the feature is, but how it helps. So biconcave disc shape to increase surface area to volume ratio for faster diffusion. That's just worth 1 mark. Hemoglobin to bind to oxygen is 1, and no nucleus to create more space for the hemoglobin, also 1. Then for question 3, we've got to state 2 of these adaptations of our sperm cell, any 2 of these, having lots of mitochondria, a flagellum and an acrosome. So add up your scores and give yourself a total. The first thing we need to do today then is understand what we're talking about when we refer to a stem cell. And quite simply, a stem cell is an undifferentiated cell. Now, This question does come up on your exam papers quite a bit, where they just ask you what is a stem cell, and the simple answer is an undifferentiated cell. That's all you need to write. Now, the key thing about these stem cells then is that they can divide by mitosis, and we've already looked at that process of mitosis in our previous lesson, and they can then differentiate into any type of specialized cell. So it's from these stem cells that we then make all of the other cell types within an organism's body. Make sure you've got those 2 points written down in your book. I'll just give you a few more seconds to get that done. When it comes to our stem cells, we actually have two types. The first one is the one that's present in your body right now, which is the adult stem cell. Even though technically you're not adults yet, you still have adult stem cells. And the second one is the embryonic stem cell. Now, hopefully the name embryonic does tell you where these ones are coming from. They're present within an embryo. So you don't have these anymore because none of you are embryos, but in the very, very distant past when you were a little tiny embryo, then you had embryonic stem cells. So make sure you got those 2 points written in your books. So what I'd like you to do then is, in your books, I'd like you to write down these 3 points about our embryonic stem cells first of all. And I've given you little pictures of some embryos on the left hand side there. Now the key words that you need are in the red box at the bottom. So I'm going to give you a couple of minutes to get these points written down with those words filled in. Let's check those answers then. Obviously tick the ones you've got right. If you've got anything wrong, write the correct answer in. So embryonic stem cells are found in embryos, they divide by mitosis, and they can differentiate into all cell types. And that's the key thing about these. The embryonic stem cells are pretty special because they can literally become any type of cell at all. So that's the really important point about those ones. When it comes to adult stem cells then, they're slightly different. And what we've got here, same thing as before, 3 sentences for you to have a go at finishing off in your books, and the keywords are again in the red box at the bottom. So I'll give you a couple of minutes to have a go at that. So in terms of our adult stem cells, these are the ones that are found in tissues like brain, bone marrow, skin and your liver. They can differentiate into some cell types and they're used to repair damage. So obviously tick the ones you got right. If you got anything wrong, write the correct answers in. Now the key difference that we can see here between our embryonic and our adult stem cells is the number of cell types they can differentiate into. So our embryonic can go into any cell type, whereas the adult stem cells can only differentiate into some cell types. So they're more limited, if you like, in what we could potentially use them for. One thing we do need to always remember in biology is it's not just humans we're talking about. We also need to remember there's a lot of information we need to know about plants. So simple question for you to have a go at answering in your book. Do plants stop growing? Answer that in your books for me. Hopefully what we said there is no, they don't. Plants actually continue to grow throughout their entire lives in certain parts. Now, the regions of the plant that will continue growing throughout their whole life are called the meristems, and we're going to find them in very specific regions on the plant: the root tips, the buds, and the shoot tips. So make sure you've got those 3 points written down in your books for me about our plants. We need to know a few key facts about the meristem cells then. So first thing is that they're small. They've got very thin walls, small vacuoles, and no chloroplasts. So what I want you to do is, after you've written down those few points about what the stem cells have, I want you to try to explain why you think meristem cells have thin walls. Hopefully the answer we came up with there is because they've got to divide, and having a thick wall would make that a lot more problematic. So by having thin cell walls, then it's far easier for them to divide. So what we can see on this screen are just a selection of headlines about what we can use stem cells for. And what I've actually got for you is a little video just to show you some of these aspects of how we might be able to use stem cells either now or in the future.
More than 600,000 people in the UK have age-related macular degeneration. It causes central vision loss, making it blurred and distorted.
Is that one? C?
No, that's not C.
That wasn't C, that was a D.
Ellie Hussein has the wet form of AMD. Its progress can be halted with costly injections, but there's no cure.
If I have to read something, there is this little white cloud in my eye that I have to struggle sometimes. Even when I read a book, I have to, but I get on with it fine.
Under the microscope, these are the specialised eye cells being used in the pioneering treatment, originally derived from a donated early embryo smaller than a pinhead. They were seeded onto a tiny patch like this and injected into the eye of an anonymous 60-year-old female patient who'd lost her sight because of AMD.
We won't know until at least Christmas how well her vision is and how long that may be maintained. We can see that they're actually under the retina where they should be and that they appear at present to be healthy.
This trial will involve 10 patients who will be monitored for a year to assess the safety of the stem cell treatment and whether their vision improves. The scientists have spent more than a decade on this ambitious project, which, if successful, would help patients with both the wet and dry forms of AMD.
What we're hoping to do is reconstruct the macula so you don't have the disease. And this is a different way of doing this. This is the first time we feel we're actually delivering a treatment to try and cure, as it were, the disease itself.
Ellie Hussein has regular, costly injections to halt the progress of wet AMD. We don't know how much the stem cell treatment would cost in comparison or when it might be available. But if it works, it would be a stunning medical advance.
So when we're talking about stem cell research, then in most cases we're going to be using these embryonic stem cells. And we're going to get these from human embryos that are about 4 or 5 days old. So 4 or 5 days after fertilisation, we've got these embryos with a number of cells, and they're the ones we're generally using. And the place we're going to get these from are spares from IVF. And you can learn more about IVF in B3 and the actual process. But the basic of it is that you're going to end up with way more actual embryos than you really need. So what some women choose to do is donate those to stem cell research when they've actually used all the ones that they wish. And they're the ones that can then go into all of these different potential treatments. So you can see in that little diagram in the middle there, all the different ways that we could potentially use stem cells in the future. So what I'd like you to do then, thinking about both the positives and the negatives, so pros and cons, I want you to write a balanced argument for if we should use stem cell research and stem cell treatments. So thinking about all the different things we can use them for, thinking about why people might be against this, then write me a balanced argument. So you give the reasons for, the reasons against, and then finally you say what you think, your final view. And your view is never going to be wrong at the end of the day because it's what you believe. So as long as you've backed it up using those ideas, absolutely fine. So I'm going to give you a good kind of 5 minutes to have a go at this. Last thing for today then is the plenary. So have a go at doing the 2 columns on the left if your target grade is up to a grade 5, and the 2 columns on the right if it's over a grade 5. I'll leave this up here for about 3 minutes, but if you think you're going to need longer, obviously feel free to pause the video. As always, you can find the answers to the plenary over on my website, scienceright.weebly.com. You'll also be able to find that under the Internet tab. Just select the week that we're in, and then you'll be able to find things like the lesson PowerPoints, the answers to the plenary, any additional work, and if there's any little experiments I thought you could do at home, then they're there too. So have a look and get using that.